he's no longer biologically useful. So a protein loses its shape, it most likely can't do its job. Occasionally a protein will be renatured after being denatured, but this doesn't always happen. It can't always happen for certain enzymes and proteins, but sometimes, occasionally, it'll take place. So now it's time to look at the fourth and final macromolecule group, nucleic acids. Now, you're probably like, what in the world are those? Well, to put it simply, they are DNA and RNA. Now, this is pretty important because DNA and RNA are blueprints for life. Without DNA and RNA, all life on Earth wouldn't have a blueprint. So anyways, let's look at the structure of DNA and RNA, starting off with the polymers and the monomers. The monomers of nucleic acids are nucleotides. So what is a nucleotide? Well, they contain 3 key ingredients. And this is a phosphate and a sugar that contains 5 carbon, an organic molecule. So it's going to look something kind of like this. It's going to be a really bad representation. So we have our little sugar there, and the little circle there is our phosphate group. And that forms the backbone of nucleic acids, which then bond to other nucleic acids and form the backbones of the polymers, these form the backbones of nucleotides, and they bond together to form the backbone of nucleic acids. Sorry about that. But there's a third key ingredient, and that's a nitrogenous base. So these bases are connected out here on the inside of our little guy here. And there are 5 different kinds of nitrogenous bases. Let me quick draw just a simple one. There you go. There's your nitrogenous base, the 2 little shapes there that are connected to each other. So, now, these 5 different kinds are called cytosine, guanine, thymine, adenine, and uracil. So now that we know this, we're going to look into the shape of DNA. DNA forms a double helix structure. You've probably seen this in any sci-fi movie or science documentary, just as like some cool animation that takes place at some point. So that's a double helix right there on the side. And so this DNA's double helix looks like this. Gotta add the nitrogenous bases in the middle. That's what it looks like, that guy right there. So, when you look at that, you're gonna first notice the outsides, and we want to know, what are those spirals made of? Those are the backbones, after all, of the double helix. That's what they're called. Well, that's our sugar and our phosphate. So that all through there is just tons and tons and tons of that circled part right here. That's a ton of those. And then you're probably thinking, so then if we know what those backbones are made of, what are those bars in the middle, those connecting things? Well, those would be the nitrogenous bases. And this is where DNA and RNA too get really, really interesting. So these bars, these nitrogenous bases, they aren't bars, but they look like bars in faraway double helix models, are connected by hydrogen bonds. And if you know what one side of these nitrogenous bases are, since there's 2 in each of these connections, so here I'm going to draw a little line in each, so that we know that it's 2 separate nitrogenous bases connecting it on either side. If you know what one side is, what the lettering sequence is, and the lettering sequence is just the first letter of each of the different bases, so like you might have, for instance, ATATCAG or CACG CTAT. So that's just the first letter of each one of our nitrogenous bases. So here's the really, really cool thing. Because we know what that one side is, we're going to know what the other nitrogenous bases are because of this certain rule with DNA. And that's this. Cytosine will only bond to guanine, and thymine only bonds to adenine. So since we know that this one might be AT-AT-C-A-C-G-C-T-A-T, well then the other side would be T-A-T-A-G-T-G-C-G-A-T-A. Get it? So, we're going to move on to RNA now. Now, RNA is almost identical to DNA, except for a few little things. One, it's only one-sided. So, it's not a double helix. It would be more like if you just took half of our double helix friend, and that's all it is. So, I'm going to have something a little more like this. That is RNA over on the side. So the second really neat thing about RNA is that it only codes for one gene. A sequence of DNA, double helix, will code for multiple different genes. RNA just codes for one. The third thing is it doesn't contain thymine. In its place, we have uracil. Remember him? So now we're still going to have cytosine bonding to guanine, but now adenine is going to bond to uracil. And then number 4, in the backbone of RNA, so in that big line part there, not the bars, the other part, in its backbone, there is an extra oxygen present in each of the 5 carbon sugars. So in that little guy I circled earlier, in the more hexagon-like part, there's going to be an extra oxygen in there. So, now that we have gone through the structure of DNA and RNA, we're going to look into what they do respectively. DNA codes for the structure of all living organisms. It is the blueprint for living organisms. It tells all the different parts of the body how to arrange themselves. So what about RNA? Well, RNA does almost the same thing, except it codes for proteins. The neat thing is the way these all interact. DNA directs RNA creation. So DNA tells your cells how to create RNA. And then RNA tells your cells how to create proteins. So it goes DNA, RNA, protein. Simple. So that's macromolecules in a nutshell. Now, they're a little complicated, so they take some time to fully understand. I don't fully understand them yet, but just keeping in mind what you've just learned is a great start. Just remember the 4 types, their basic structure, how they function, and how they're connected to one another. And you should have a good groundwork laid for your education on macromolecules. Before I end this video, I would like to thank a couple really good resources that I used. Britannica.com, or Britannica, yeah, said that wrong. They're a really good source of information. The Amoeba Sisters does a really, really good video on biomolecules, such as macromolecules. Mr. Sadler and his macromolecule slideshow was extremely helpful in this. Crash Course with all of their videos on biology and biochemistry is extremely helpful. CK12.org and materialsworldmodules.org.